Hi, this is Les and Amy from the RevGenetics Podcast. You can listen to the podcast on YouTube and now on Spotify, where we do a deep dive on supplements and longevity studies.
And today, we are taking one of our most fascinating deep dives, I think. We're bridging the gap between, well, social science and molecular biology.
We're exploring how the stressors of the outside world, your neighborhood, your income, your environment, actually change the biology inside your cells.
Yeah. Our source material is a comprehensive review article focusing on P53-dependent cellular senescence in health disparities.
Okay, that title sounds dense.
It does. But the mission here, the question we're tackling is so important, we're looking at a major puzzle in public health.
And the puzzle is this. Why do some populations, people facing, say, constant financial stress or high levels of pollution, consistently seem to age faster?
Exactly. Why do they get major chronic illnesses, things like heart disease, diabetes, even some cancers, so much earlier than people who are better off?
And it's not just about lifestyle choices or healthcare access, though those matter. This research goes so much deeper.
It does. The revelation here is that social adversity isn't just in your head. It's not just psychologically damaging. It acts like a literal toxin at the molecular level.
So this deep dive is going to explore the actual cellular switch that this social stress flips, accelerating our biological age.
It's a powerful connection. And to understand how our social life can shorten our biological life, we really have to start inside the cell. We need to talk about cellular senescence.
Okay, let's unpack that. And we can use a really simple analogy. Think of your body as a big factory. Your cells are the workers.
Mm-hmm.
Cellular senescence is basically when a cell, just permanently retires. It gets too damaged, maybe its DNA gets fried, and it just stops dividing forever.
And that permanent stop is initially a really good thing. It's a critical failsafe. Think of it like a quality control manager pulling a faulty product off the line.
So it's a protective mechanism.
Absolutely. If that damaged cell were allowed to just keep dividing, it could grow uncontrollably. And that's how you get a tumor. That's cancer. So this retirement is good. It protects you.
But if it's a failsafe, how does it become a part of the problem, the accelerated aging problem?
Because these retired cells, they don't just disappear, they get stuck in the tissue. And this is where a key molecule, P53, comes into play.
Okay, P53.
You can think of P53 as the cell's internal damage inspector, or the factory supervisor.
The supervisor who checks for damage on the assembly line.
Precisely. When a cell suffers serious damage from toxins, radiation, even just severe internal stress, P53 gets activated. It looks at the damage, and if it decides the cell can't be repaired,
It flips the retirement switch.
it flips the switch.
Wow.
It locks that cell into that permanent senescent state.
So P53 is the good guy here, initiating this protective retirement. Why then do these retired cells start causing trouble?
Because once they're retired, they develop a really nasty habit. It's called the SASP.
The SASP, okay, what's that?
It stands for senescence-associated secretory phenotype. But basically, the retired cell starts leaking this cocktail of toxic inflammatory chemicals all over its neighbors.
So they're not quiet retirees. They're complaining and causing problems for everyone else in the factory.
That's the perfect analogy. And those chemicals are the real drivers of disease. They're powerful inflammatory signals, things like IL-6 and TNF-alpha, and they create this chronic, low-grade inflammation throughout your body.
And that kind of inflammation is at the root of almost every age-related disease we worry about.
Yeah.
Heart disease, diabetes.
You got it. The SSP damages neighboring tissues. It causes plaque buildup in arteries. It makes your muscles resistant to insulin.
So what's the big connection here? What flips that retirement switch early?
That's the key. The chronic stress and the toxins found in disadvantaged environments. They cause more of that DNA damage. They activate that P53 inspector much earlier and much more often.
So their cellular retirement schedule gets pushed way up. Their biological age starts to outpace their actual age.
That's it, exactly. Now that we've got the internal mechanism down, let's look at the external world, the specific triggers that are activating P53 too early.
So where does the paper draw the clearest lines between the environment and this cellular damage?
Well, the sources point to a few major culprits. First up is direct environmental toxins, the things we breathe. Urban air pollution is a huge one.
We usually think of air pollution and, asthma, lung problems.
Right.
But the damage is much wider.
Oh, much wider. When you inhale that ultrafine particulate matter, it doesn't just stay in your lungs. It causes a massive burst of reactive oxygen species. We call it oxidative stress.
Oxidative stress, right. That's like internal rusting.
A great way to put it. And that rest directly damages cellular parts, including the DNA. And as soon as DNA is damaged, P53 is on the scene.
So living in a polluted city is like giving your cells a constant low-dose bath of DNA-damaging acid.
That's a very vivid and accurate way to see it. And it's the same mechanism with things like tobacco smoke. The paper points out that smoke is a powerful trigger for this process in lung cells.
Which would explain why diseases like COPD can be so much worse in certain communities.
Yes. But beyond physical toxins, the most fascinating part of this for me is the link to pure psychosocial stress, financial worries, discrimination, that kind of thing.
How does something like worrying about rent turn into a physical signal that damages a cell?
It's all mediated by our stress hormones, especially cortisol. The research talks about something called the weathering hypothesis, the idea that constant stress just wears your body down over time.
And John Henryism, right? The idea of this high-effort struggle against the odds.
Exactly. When you're in that state constantly, your body is always releasing stress hormones. It's not a short burst of fight or flight. It's a sustained, years-long activation.
And that's what does the damage.
Yes. That sustained activation is terrible for our mitochondria, the cell's powerhouses. It causes them to malfunction, which creates even more internal oxidative stress, or rust. The cell basically reads that chronic worry as a physical injury.
Which then engages the P53 pathway.
And pushes the cell toward early retirement. So the stress is not abstract at all. It is physically damaging the machinery inside your cells.
That's a profound connection.
Yeah.
It really changes how you look at health and social equity.
It does. And then you have to add the metabolic link to that burden. Disadvantaged areas are often food deserts. Access to healthy food is a real problem.
And we know obesity itself is a huge accelerant for senescence.
A massive one. Obesity creates this state of chronic metabolic stress, and that stress causes premature senescence, especially in fat tissue and the liver.
And I bet those senescent fat cells are really problematic.
They're some of the worst offenders. They become these little factories for SASP factors, pumping out inflammation.
So you get this vicious cycle. The inflammation from the senescent cells makes the metabolic problems worse.
Which creates more stress and more senescent cells. It's a horrible feedback loop. And that's why you see diseases like type 2 diabetes and heart disease not just appearing earlier, but also being more aggressive in these populations. There's a real senescence gap.
So the key takeaway here is that these disparities aren't because of some intrinsic biological difference. They are the direct result of unequal exposure to things that literally speed up the normal aging process.
And that makes the solution much clearer. If the problem is too many toxic old cells, the intervention has to be about either getting rid of them or shutting them up.
Which brings us to the exciting part, sanotherapeutics. This is the biological side of what the paper calls the gero-equity framework.
Right. And there are two main categories of these drugs. The first one is senolytics.
Senolytic. So senolytic means to kill or destroy.
Exactly. They are designed to selectively kill those retired, toxic, senescent cells. It's like sending in a specialized cleanup crew to remove the troublemakers from the factory floor.
So what are the leading candidates here?
Well, the most famous combo is dasatinib plus quercetin, or D plus Q. There have been some early human trials that showed it can reduce those circulating SASP factors.
Interesting. Any others?
Yeah, fisetin is another one. It's a flavonoid found in things like strawberries. It's a natural senolytic. And then there are more powerful drugs like navitoclax.
Okay, so those are the killers. What about the second approach? Xenomorphics. That sounds a bit gentler.
It is a softer touch. Xenomorphics don't kill the cell. They just modify it. They quiet down that toxic SASP leak. They basically tell the retired cell to stop causing trouble.
And are there drugs we already use that do this?
Absolutely. The best example is metformin. It's one of the most common drugs for type 2 diabetes, and it works, in part, by suppressing the SASP.
Wow. And what about rapamycin? We've talked about that one before.
Rapamycin is another major player. It works through the mTOR pathway, and it helps guide damaged cells toward a reversible resting state called quiescence, instead of that permanent harmful senescence.
RevGenetics is helping us unpack today's topic, thanks to them.
We're grateful that they support deep dives into geroscience and the molecular foundations of healthy aging, which often involves understanding how key pathways like AMPK and mTOR can be optimized.
Okay, so these drugs are incredibly promising. But the review makes a really strong point that senolytics alone are just a temporary fix.
Right.
If we keep sending people back into the same environments that caused the problem.
Absolutely. This is the other half of the gero-equity solution, the policy half. The drugs are essential, but you have to pair them with public health strategies that reduce those upstream stressors.
You have to stop the damage from happening in the first place.
Precisely. We're talking about things like environmental justice. If air pollution is accelerating senescence, then fighting for cleaner air in vulnerable neighborhoods is a direct form of senescence prevention.
You're treating the root cause, not just the symptom. And I imagine addressing things like financial stability fits in here too, to lower that chronic stress.
Yes. Things like anti-poverty programs, stable housing. They directly lower the amount of circulating stress hormones. You're reducing the wear and tear. In a very real way, structural safety creates cellular safety.
And what about on a personal level? Lifestyle changes.
Oh, definitely. The sources confirm that regular exercise basically acts as a natural senolytic. It helps the immune system clear out senescent cells more efficiently.
And diet.
Antioxidant-rich diets reduce the initial DNA damage, which means the P53 supervisor doesn't have to pull the retirement lever as often.
So when a city builds a new park or a grocery store with fresh food, they're actually rolling out a large-scale anti-senescence intervention.
That is the ultimate conclusion here. It makes the biological case for social reform completely undeniable. The fight for health equity is a fight for cellular resilience.
So before we wrap up, what's next? What does the research community still need to do to really nail this down?
The biggest gap right now is the need for more long-term studies. We need to follow large, diverse groups of people for decades.
And measure not just their health outcomes, but their cellular health too.
Exactly. We need to measure specific senescence biomarkers along the way. Things like the P16 protein in blood cells or the actual levels of those toxic SASP chemicals.
So you can draw a direct line from, say, living near a highway for 20 years to having elevated senescence markers, and then to developing heart disease 5 years earlier.
That's the gold standard of evidence. And we need that evidence to really integrate the biology with the policy to create solutions that actually work for everyone.
And to make sure that when these new senolytic drugs are tested, they're tested on the populations that need them most.
Yes. The trials have to be inclusive. They have to account for these environmental factors to make sure the treatments are safe and effective for the people who are experiencing this accelerated aging.
I think the final takeaway here is just so powerful, and it goes way beyond just taking a supplement or a new pill. The most effective path to healthier aging for all of us really requires tackling these big structural problems: clean air, safe housing, food security, that are forcing our cells to get old before their time.
Until we fix the environments that create the damage, we're just fighting a losing battle against the symptoms.
Yeah.
We have to stop the external forces from triggering the internal decay.
We hope you found today's podcast on how social status and environment accelerate cellular aging informative.
Thank you for listening to the RevGenetics Podcast.
RevGenetics.com for the best longevity supplements on Earth.